Have you ever taken a drug for an illness and wondered, what is this drug actually doing? In many cases, that drug is either activating or inhibiting a specific enzyme in order to slow down or speed up a reaction in your body. Enzyme function is so key for our body's processes that often when you have an enzyme that might be running faster or slower than it normally would, you will have a disease state develop. In this lecture, I want to talk specifically about what these different mechanisms are for changing the rate of our enzyme reactions. And we'll also take a quantitative look at how we can calculate the impact of those drug molecules or other signaling molecules that are used to change your enzyme activity. So in our previous lecture, we've shown how we can calculate the rates of our reactions using the Michaelis-Menten equation. So we have a couple key constants here. Our Vmax, that represents the maximum rate for a specific concentration of our enzyme. The Km, that is a combination of our rate constants and can also give us an approximate value of the affinity for the substrate to our enzyme. And then, of course, we have our variables of the substrate concentration and the reaction rate based on that substrate concentration. So this is the ideal scenario. Now, this ideal, of course, can deviate in several different ways. What we're mostly going to be concerned about is the presence of inhibitors that will change that ideal rate. Enzyme inhibitors can take a variety of different forms. The first possibility is environmental factors, such as our pH or the temperature. As we've seen in our earlier lectures, the structures of our proteins are strongly affected by pH and temperature. And if we change that structure, it's going to change the interaction between our enzyme and the substrate and potentially change the mechanism that we might be trying to use in order to catalyze our reaction. So for any given enzyme, you'll see one optimum pH. So you can see we have two different enzymes here, glucose 6-phosphate with an optimum pH just below 8. And we have pepsin here with an optimum pH just below pH 2. So pepsin is designed to work in the stomach, where we have very low acidity or a very high degree of acidity and a low pH. And our glucose 6-phosphatase is designed to work in our cells, where we tend to have a higher pH. So we have our buffered pH. It's going to be around 7.4, right near the optimum of our glucose 6-phosphatase. Now, of course, you should also note here, so one of the reasons why pH has such a large effect is because it can affect many of those key amino acids that we might be using to help catalyze our reaction. For example, if you have an acid-base catalyst mechanism, any change in pH is going to change the protonation state of your active amino acids and can then ruin that catalytic mechanism. This sort of inhibition mechanism is a bit different from the initial drug example that I provided, because it's not really a specific molecule binding to our enzyme and then slowing down our reaction. It's just an overall kind of change in our protein structure caused by environmental conditions. So most of what I'll be focusing on today is actually small molecules binding to our proteins and changing protein activity in that way. These small molecules that affect our enzyme activity are called inhibitors. So an inhibitor is a compound that will decrease an enzyme's activity. We have several different kinds of inhibitors that we commonly see. There are irreversible inhibitors or inactivators that will actually react with the enzyme and prevent the enzyme from catalyzing its target reaction. So these are some of the most powerful inhibitors. So they can be extremely toxic because you can completely shut off a particular reaction, but they can also be used as drugs as well. They would just be more potent drugs that could then also be potentially dangerous as well. Now, it's important to note here that just because it's an irreversible inhibitor doesn't mean it's going to completely stop our reaction, right? So you could have if you only add a small amount of that irreversible inhibitor, you will only inhibit some portion of your enzyme. You might have some other enzymes that are still active. Or, of course, even if you add enough of that drug in order to shut down every enzyme, you still will have more enzymes being produced by the body to try to continue that reaction. So it doesn't necessarily completely stop a specific reaction. It would just slow it down. Now, the other type of inhibitor would be a reversible inhibitor. So this, of course, as the name suggests, can be reversed. So it can bind to our enzyme, but then it can dissociate at another time in order to relieve that inhibition. So often you might have a reversible inhibitor that's a structural analog of a specific substrate or the product. So it looks similar, so then it might bind into the same site and kind of compete with the substrate. These would then be used to try to slow down a specific enzyme by adding kind of a competition effect, but they wouldn't permanently stop that enzyme activity. Now, of course, a reversible inhibitor doesn't have to be a structural analog that looks similar to the substrate. It could also just be a small molecule that binds to a different site in our protein and changes the conformation of our protein to affect its reaction. So this would be more of an allosteric mechanism, right, where it's binding to a different binding site and then affecting the interaction at our main binding site. Let's focus on our reversible inhibitors first. So there are a couple different kinds of reversible inhibitors, and these are determined based on their specific mechanism for inhibition. So I want to look at the actual quantitative effect as well as the theoretical effect here. So first we have a competitive inhibitor. So this is something that would be a more structural analog of our substrate. So it looks similar to our substrate. You can see that marked down here. We have our inhibitor that has kind of the same binding face as our substrate, right? They both have kind of that pointed binding site that then fit into the binding site of our enzyme. So we could bind either our substrate or our inhibitor, but not both at the same time, since they both occupy that binding site. So in terms of our binding model, that's represented over here on the left. We have two different equilibriums here. We could either bind with our inhibitor to form an enzyme inhibitor complex, or we could bind with our substrate and form the enzyme substrate complex. Of course, it's only if our substrate binds that we can actually react and create our product. So any time that our enzyme spends bound in this enzyme inhibitor complex, we won't be producing product and will slow down our reaction. So how would this inhibitor affect the rate of our reaction? Of course, as you see, what's happening here is that our inhibitor is effectively slowing down the binding of our substrate to our enzyme, right? Instead of the substrate just being able to approach and bind to our enzyme, we have to wait for our inhibitor to be released from that enzyme so that then we have free enzyme available for our substrate to bind, right? So you can see where the competitive term comes in, because both our substrate and our inhibitor are both competing to try to bind to that single binding site. So essentially what we're doing here is we're decreasing the affinity of our enzyme for our substrate. Or since our affinity is kind of expressed in form of our Km, we get an apparent increase in our Km value, right? Essentially, our affinity is decreasing, which would be an increase in our Kd or our Km, because of this competition with our inhibitor. So if we look at the binding constant for our inhibitor, so this would be the Ki is referring to the equilibrium constant for our inhibitor binding to our enzyme. This is now going to directly affect our enzyme kinetics itself, because this is determining how much our affinity is decreased for the substrate. So we take this Ki and we calculate an alpha term based on the inhibitor concentration as well as the Ki, because as we know, the concentration of our inhibitor determines how much of that inhibitor will be bound to our binding sites, right? The bound protein fraction, essentially. And now we use that alpha term to modify our Km, right? So the true Km of the protein remains the same. Our enzyme isn't changed by that interaction with the inhibitor. It's just that our inhibitor is slowing that down depending on the strength of the binding interaction between our inhibitor and the enzyme. So our Km stays the same, but it's now modified by this alpha term within our Michaelis-Menten equation. Now, of course, at the same time, our Vmax is not changed because, right, there's still some point where we reach a maximum possible velocity where all of our substrate is bound in our enzyme, right? We haven't changed the rate if all of our substrate could bind. We've just changed the rate at which our substrate does bind because we have this inhibitor here, right? If we had a super high substrate concentration such that it managed to completely overwhelm the inhibitor, our Vmax would still be the same. So if we look at the kinetics of this sort of inhibition using a Lineweaver-Burke graph, what we would see is that our lines would intersect at the y-axis. Let's go ahead and look at our graph directly here. So here we have our typical Lineweaver-Burke graph with our double reciprocal of the substrate concentration and the initial rate. And we have our enzyme activity plotted with different inhibitor concentrations. So with no inhibitor, our alpha term simplifies to 1. Right? So remember, alpha equals 1 plus the inhibitor concentration over the Ki, the binding constant for our inhibitor. And so if our inhibitor concentration is 0, this just simplifies to 1. So here's our original enzyme. You see we would have a slope and a Vmax determined by our y-intercept here. That would be, of course, our original Km and Vmax for the protein. As we increase the inhibitor concentration, our alpha value also increases, right? So this is increasing depending on what our Ki is. We'll determine that overall alpha value. And you notice, so our Km, or the apparent Km, is changing, right? Because we're changing the relative affinity by having this competition with our competitive inhibitor. So the slope then changes, since the slope includes our alpha and our Km component. But as I just mentioned, the Vmax does not change. So our y-intercept stays the same at all times. And we get this sort of graph where as we change our inhibitor concentration, the slope is always changing, but the y-intercept, the Vmax value, always stays the same. So again, we see how that Lineweaver-Burke graph would allow us to quickly see what kind of inhibitor we're dealing with if we have a competitive inhibitor present with our enzyme. So what about other kinds of reversible inhibitors? So the other main kind of reversible inhibitor would be an uncompetitive inhibition. So this is more of the allosteric mechanism of inhibition, where it doesn't bind to the same binding site, but instead it binds somewhere else and inhibits the catalytic function of our enzyme. So in this case, we have our substrate with that typical shape to fit into our binding pocket. So our substrate binds here, but now we have a separate binding site that fits our inhibitor. Our inhibitor would bind there, and we could form an enzyme substrate inhibitor complex with both of those molecules bound in our enzyme. So again, we have a new equilibrium to consider. So of course, for our original enzyme model, we have our enzyme substrate binding equilibrium, which would then react to produce our product. But in this case, now we have our inhibitor that can bind to our enzyme substrate complex, and we form this inactive enzyme substrate inhibitor complex. So we have another Ki here that's relating to the binding constant of our inhibitor. In this case, we put a little apostrophe. So this is Ki prime in order to indicate that this is a different binding than our competitive inhibitor that would be binding to the main substrate binding site. So how does this affect our kinetics? So again, we're not directly affecting the binding of our substrate to our enzyme. Instead, we're going to be affecting the maximum rate, right? We're slowing down the actual rate of our enzyme in that enzyme substrate complex. So we're again adding a new term, an alpha prime, to our Michaelis-Menten equation. But you'll notice it's over here with our substrate concentration instead of with our Km. Again, we have our alpha values and our Ki's defined in the same way. So there's no change in our definitions here, except for where that term pops up in our Michaelis-Menten equation. So the end result is that we would have a decrease in our Vmax and an apparent decrease in our Km, but there would be no change in our Km Vmax ratio. And this would result in a Lineweaver-Burk graph with parallel lines. Now, this will make a lot more sense if we look at the graph directly. So if we take the double reciprocal of our Michaelis-Menten equation with our modified term, you'll notice that the alpha term now ends up over here on our y-intercept, right? This is why our apparent Vmax value starts to change, right? Because alpha over Vmax is now changing as we change the inhibitor concentration. And so if we looked at each of these graphs, it would look like they each have a different Vmax. Again, of course, the true Vmax is not changing. It's just that the presence of inhibitor is temporarily altering that Vmax in the presence of our inhibitor. You'll also notice that our slopes here are the same, right? These are our parallel lines, which of course makes sense from our equation. Our slope is here as the Km over Vmax, and there's no alpha term here. So as we change the inhibitor concentration and change our alpha value, our slope still stays the same. Now, of course, since our Vmax is changing as the y-intercept moves, our Km actually is also changing, our apparent Km that is, because our x-intercept is also changing here. So it's only the slope that stays the same. Our apparent Vmax and our apparent Km are both changing here. So again, we see a very simple method by which we could determine an uncompetitive inhibitor. If we see parallel lines as we change the inhibitor concentration, we have an uncompetitive inhibitor. Now, as with most questions in biochemistry, it's often not just one simple answer of either competitive or uncompetitive. We have a lot of inhibitors that will be mixed inhibitors, where they can bind to the enzyme with or without the substrate, and they can influence both the binding of the substrate as well as the activity of the enzyme substrate complex. So with the mixed inhibitor, you have, it can bind to our free enzyme, and we would have an equilibrium constant for that process. But our substrate can still bind here as well, and we can get an enzyme substrate complex where our inhibitor can also bind with a separate binding constant. So now we have even more equilibriums to deal with it binding either without our substrate or with our substrate in two different processes. So here, this is represented by two binding sites that are very close to each other. So our inhibitor is affecting the interaction with our substrate, but it's not necessarily completely blocking it the same way that was in our competitive inhibition. This sort of mixed inhibition essentially acts as a combination of competitive and uncompetitive inhibition in terms of our quantitative calculations. So now we have an alpha term with our Km and an alpha prime term with our substrate. And we'll have to use both of these to calculate the overall rate of our reaction. So now we would see a decrease in our Vmax, as we saw with our uncompetitive inhibition, and an apparent change in our Km, as we saw with our competitive inhibition. In our Lineweaver-Burke graph, we would see our lines intersecting left from the y-axis. So we would still have an intersection, but it would no longer be on the y-axis, as our Vmax values would be changing. There is a special case of mixed inhibition that we refer to as a non-competitive inhibitor. This is a mixed inhibitor where there's no change in Km. So this would occur when both of our alpha values are identical, or in other words, both of our binding constants here are the same. So this would say that the binding of our inhibitor is completely unaffected by our substrate, right? It binds the same whether the substrate is absent and we have the free enzyme, or whether the substrate is present and we have the enzyme substrate complex. So it's just a special case of our mixed inhibition. Let's go back to our Lineweaver-Burke graph. So again, we take a double reciprocal of our Michaelis-Menten equation. We have our alpha term here that's modifying our slope. So notice the difference in slopes between our lines that then leads to our intersection point. And of course, we have an alpha prime term that's modifying our y-intercept. So notice our y-intercept and our apparent Vmax value are changing here for each of our different inhibitor concentrations. And of course, at the same time, our apparent Km value is changing, and our slope of that Km over Vmax ratio is also changing here. So you see how the inhibitor is increasing the steepness of our slope. So mixed inhibition, of course, can be a bit more complicated, but again, notice that we can easily identify this just from our graph, our Lineweaver-Burke graph with changing inhibitor concentrations. We would just look for the change in slope and in our intercept point, with it now having varying intercept points and our intersection occurring to the left of the axis. So for each of those reversible inhibition mechanisms, we had a transient interaction between an inhibitor and our enzyme. And again, as we saw, it could be competitive or uncompetitive, where it's binding at the main binding site or a separate binding site, but it's still a association process there, where it can bind, but then it can unbind with a resulting binding equilibrium there. Let's compare that to our irreversible inhibition. So an irreversible inhibition, again, would require our enzyme to be chemically modified in order to prevent our reaction. A good example of this is in our chymotrypsin enzyme that helps to digest proteins by cutting up our peptide bonds. So our chymotrypsin has a key serine in the reactive site, shown here, and our inhibitor here, DIFP, uses a fluorine to react with our hydroxyl group of our serine and add on this large structure to our serine. So you can see now we've created a covalent bond between our serine and this phosphate from our inhibitor. Now, if we look at the active site of our chymotrypsin, so normally our serine here is helping to interact with our substrate and stabilize it within our enzyme. It's also actually involved in our mechanism where our serine hydrogen bonds to our transition state and helps to stabilize this negatively charged oxygen. So our serine is essential for stabilizing our transition state and allowing our reaction to proceed at a faster rate. If we attach that large DIFP group here, that's going to completely block this active site and prevent our enzyme from functioning normally. So our serine would no longer be able to stabilize our transition state. In fact, instead, we'd have a large molecule sitting here and actually blocking our substrate binding. Now, again, remember, since we actually formed a covalent bond here between our serine and our inhibitor, there's no way that we can easily remove that inhibitor. We'd have to actually react and break that covalent bond in order for our enzyme to work again. So for the most part, it's going to be irreversible. Our enzyme will just end up getting broken down, and we'd have to produce a new enzyme to catalyze that reaction to replace our inhibited enzyme. So this shows us just a small taste of the different mechanisms that we can use in order to alter the activity of our enzymes. Again, all of these are important mechanisms that drug molecules use to slow down or even to speed up rates of reactions. So we've mostly focused on the inhibition side of things, but you could also activate reactions through similar mechanisms as well. And again, controlling the rates of those reactions is incredibly important to make sure that our body behaves as it's supposed to, right? With all of our reactions proceeding at appropriate rates and then producing the right amount of molecules for our body's use.